So the human body is built out of many, many structures called cells, and every cell has its own unique shape. And the function of those cells is very dependent upon the shape that the cell has. And so one way to think about this, what the design challenge is for each cell in the body is imagine that you were challenged with building a house, but your house had to do two things. It had to be able to withstand the next hurricane, and at the same time very dynamically remodel and divide into two houses. And so what we study are the dynamic machinery that allows a cell to carry out those two very dichotomous roles. And what we have found are a whole host of proteins, which are the building blocks inside the cell, that have to be restructured, reorganized, assembled, put together in order to create these various shapes. To approach our studies, we've always used a very simplified model organism called Dictyostelium. It's a very simple protozoan that has a genome and a makeup very similar to ours, but it's much simpler. So they have fewer versions of every protein. And by studying that, we can identify elements or molecules, proteins that the cell will use to carry out these shape change events. And then what we do is we look to see how the concepts that we've learned in our model system then apply to a human disease. One disease that we are investing quite a lot of time in right now is pancreatic cancer. And what we've learned is that the major mutations in a person's DNA that are causative, that cause you to develop pancreatic cancer, turn out to be mutations in the same kinds of proteins that are regulating much of the machinery that confers shape of the cell. And so by using this sort of integrative approach where we go from a simple model system to something really complicated like a human disease, we're able to come up with unique insights and maybe even ultimately, we hope, therapeutic strategies for treating those diseases.